Without further ado, let's welcome Professor Fischer for a talk.
Okay, good morning. First of all, a very warm thank you to Professor Ashish Sen and his team who've been working round the clock so hard to make everything run so perfectly. And thank you for being here. I'd like to talk about a topic that we've been interested in for a few years now, not that long, but it's about using ultrasound in conjunction with liquids and materials. And we're based in Heidelberg, both at the Max Planck Institute for Medical Research and the university. Today I will talk about work that is primarily done by three postdocs, Tanasi, Nicolas, and Kai. And these are our funding sources, and this is the current group of PhDs and postdocs in my lab. So I'd also like to thank a collaborator for this project today, which is Albert Pottinger at the TU Eindhoven. We're all familiar with 3D printing. Most of us have such a device in the lab. And what is done is very often extrusion printing. A fluid is mixed with some material and ink, and then it's positioned somewhere. And so this requires a pressure, it requires flow, and it's typically a serial process where you position things one point at a time, and therefore generally is quite a slow process. Now it's also used in biofabrication. People use bioinks, and they print cells, and they even try to get organoid type structures printed. It's typically layer by layer or point by point, and therefore requires that your sensitive biological material is kind of pressed through a nozzle, and it takes the time for making the structure. Now nature does it differently. Nature doesn't print. Nature assembles things, and so here the material basically will fall into place by usually biomechanical mechanisms. It's entirely parallel. Everything happens at the same time, and therefore it's a volumetric process that's very, very fast. And so what I'd like to ask today is whether we could build something or a mechanism to also achieve volumetric fabrication, and can this be experimentally realized by a nice method? So this is not like this has been, nobody's ever thought about it, has quite some history, and of course one aspect that has been touching on this is the question of self-assembly. And there was a DARPA project funded a couple of years back where people were imagining that there is this magic colloidal suspension, the claytronic, so it's some clay, so basically some simple material, tronic, meaning it probably has an electronic or computational feature, and this project was imagining that you could have this matter, and it wouldn't just be what we know as of dead matter, but it would be in a way that these particles or structures could come together and magically form the object that you want. Now, this, I think, has not resulted in anything experimentally realistic. This has mainly been driven by computer scientists, and it's very easy to make the computer simulation, but how do you do this in an experiment, right? How do you bring things in space in the same time and location, and maybe even that it has a function? And so the question is, how do we do assembly? And how do we do this faster than 3D printing? And we could be maybe thinking about gravity, which sure would work. If you could shape gravity, things would fall into place, but it's not an experimentally accessible parameter, so let's go to fields. One might think that you use magnetic fields or electric fields to push things, but the problem is you cannot generate stable trapping points outside of having either current-carrying wires or charges. So you cannot project an electric field shape into space and make it an object where you can trap something. So the problem is you need these current-carrying wires and you need charges, so therefore you would have to already build a structure basically to attract this material. So not so good. Light, of course, is being used. Many printing methods are based on light. The problem with the light is that, yes, you can, to some extent, shape it. Typically, you can only realize planar type projection, not really volumetric, but you can always interact only with a light-sensitive material. So you can only structure a photosensitive polymer, if you like. You cannot take, like here, the cells and print an ear directly. And so that's what brings me to sound and pressure waves. Now, this is the recipe. I'd like to say a few words about ultrasound, and then I'd like to say how one might use it as a method for shaping pressure in fluids. So we're all familiar with ultrasound. above 10 kilohertz, which we cannot hear. The bat uses about 100 kilohertz. It sends out a simple wave, like your distance sensor in the car, and it measures when something comes back. It's a simple plane wave. And even in medical imaging, which tends to be from megahertz to 10 megahertz or so, you send out very simple waves, and the magic lies really in what you detect. You're detecting a complicated pattern that comes back, that gives you the object. But what you send in here are very simple, almost planar waves that are emitted and that enter an object. Now, ultrasound and sound in particular can also exert forces. You all know that. You will probably have an ultrasound cleaning bath at 40 kilohertz. I used to live in the Black Forest, and if you want to cut this cake here without squishing the cream, you can use an ultrasonic knife. You can weld, you can even have a haptic device. You can destroy DNA by fragmenting it. You can destroy kidney stones with shock waves. And the latest is you can have magnetic resonance guided ultrasound for high intensity focused ultrasound in surgery. So ultrasound can be used for these applications. In none of these applications is it in any way sophisticated. It's just setting up an intensity in space typically. But it can interact with matter. This is a simulation from a Japanese group. So we have a resonator, we have a speaker, and we have a reflector. And as you would expect, sending up a resonation mode, you will have regions where there is high intensity and low intensity. And if you manipulate this, you can use this potential energy surface and, for instance, trap polystyrene particles. Okay, so these are levitating polystyrene particles. You can see the highly symmetric because that's the resonator geometry. And in these nodes, depending on the material property, you can manipulate matter. So we can exert forces, we can manipulate matter, but it's all rather simple. It's very trivial. The geometries are always some resonance modes and therefore highly symmetric. You can also use it to assemble cells. The beauty, of course, is that ultrasound is very benign. You don't destroy your cells. It's all parallel, so we're not doing point by point, but we're doing it together. But again, the patterns you get are going to reflect the geometry of your resonator. And so what I'd like to ask is, can we do something more complicated? Can we get towards engineering organoids? Can we print Pac-Man with cells? Okay, and this is not going to be possible because we have not a symmetric structure that we can realize with a resonator. So we have to somehow get a method of just projecting a very simple plane wave, which will do very little in terms of a structured field that would allow us to manipulate matter in different locations. We have to get away from this, and we can take inspiration from light. We do have light projectors. So here would be, in a technical sense, a transducer, a vibrating surface that emits a wave like this, and compare that to light, where you can project. You wouldn't see this if it wouldn't project an image. So here we have a complicated pattern. And the question we want to ask is, can we take sound or pressure, and can we project it into space so that when we go into an experiment, we would be seeing, for instance, high intensity wherever we see red, and we would see no pressure intensity in the places that is white. So it wouldn't be something we see by eye, but we could measure it with something like a hydrophone. And if we could make a more complicated pressure pattern, then the idea is we can manipulate matter in a more complicated way. And one way that this has been done to some extent has been to basically increase the number of wave generators or transducers. So because as you overlap many, many waves, you get an interference, and as you get an interference and you shape all of these phases here so that they interfere, you can, for instance, get a more complicated pressure pattern. This is from my colleague in Bristol, and he's used it to basically form a bottle-type beam to manipulate one particle. So with one particle, we're not going to build our Pac-Man or whatever else. We need to make this more complicated, and also we probably need more complicated pictures. So this is not really scalable because the waves that you can form with this is rather limited. And so that brings me to the acoustic hologram. So this is our very, very simple solution to this problem. We're using a simple transducer, only one. It produces a plane wave, and on top of that, we basically take a 3D printed plate, which is the hologram. And what now happens is that as ultrasound propagates through this plate, and imagine this has different thicknesses, so at each location, the sound will traverse this plate, it'll be immersed in water, and depending on whether it goes through the plastic or the water, there are different sound speeds. So by changing the relative amount of plastic or water, you locally fix the phase that comes out. So you vary the phase. So therefore, you basically have, at each point, a unique phase. And this now allows you to generate interference patterns which are more complicated because, as you can see here, this is about a sort of 4 centimeter by 4 centimeter plate. We can work with a resolution of a 3D printer, which means we can easily work at 100 microns, 200 microns at the wavelength of this ultrasound and define at each position its phase. And so this is what my PhD student did in his project. He wants to now project a pressure pattern in the form of the peace dove in water. So this is the target image. So what we do is we have a transducer. The transducer needs to go through this hologram, which we need to build. And then the idea is by changing the phases here, the waves will interfere and form an image, which will be the pressure image somewhere in solution. And this is very simple to normal or similar to normal holography. So we're going to use an iterative approach, very similar to the Gerchberg-Saxton. The technical detail is that ultrasound is near field, and we need to take care of the angular distribution. So we use something that's called the angular spectrum method. But the idea is you have a fixed amplitude here from the transducer. You project to here. You keep all the amplitudes you like that are like this picture, and the other ones you set to 0, and then you propagate back, and that way you get a new set of phases, and you iterate this until you get this image here. Once you've computed this, you know that this phase distribution here, if you can project this phase distribution onto your ultrasound wave, it should form this image, right? And the phase here is minus pi to pi. And that means we need to put a mask in front of the ultrasound, and this we can 3D print because the height will correspond to a particular phase value. So all we need to do is take the phase, convert it into a height map, go to the 3D printer, and we print the hologram. And then the idea is if we can put this in front of the transducer, and then in water, we will get a pressure pattern. And this is the experiment that Kai did, and the point is, even with the most trivial printer, you already have something like 15,000 positions where you define the waves. This is technically impossible to do at high power with transducers. And so this is the experiment. This is in water at 2 megahertz, and you see the scale is from 0 to 40 kilopascal. And this is not an image you can see by eye, but you can take a hydrophone and you can scan it in your fluid, and at the right location you will get this. So this is probably the most sophisticated ultrasound pressure patterns or sound patterns that have ever been formed by a really, really simple method. And this, of course, this wave comes from a source, goes through the fluid and goes at the other side. So we can also ask the question whether this will work in 3D. So here is our image, our pressure image. So again, this is not something we can see by eye, but this is something where we'll have high intensity and no intensity of sound. And of course, we can follow this as it goes through the volume. And as it goes through the volume, we see this one plane where we have arranged for this image to form. So then what we can do is we can, of course, encode more information, and we could take another type of hologram, and we can, as it progresses, we can encode the first image, the second image, the third image, and the fourth image. This is one and the only same wave. So that means we have a chance to define pressure not just in one plane, but we can also define it as it is propagating through a volume. And why is this interesting? Well, I mean, until now, you need to have a very, very complicated setup to steer, for instance, ultrasound in transcranial ultrasound applications in the brain. It's really tough to have an interesting shape. It's tough to keep shaping this, so most people just focus it. And you can't really have a chance for correcting things. So these are colleagues in Spain. We've patented this idea. These are colleagues in Spain, and they have, for instance, used a hologram here to compensate for the fact that the human skull is not a very nice and smooth plane parallel surface where the transducer sits, but is really a wobbly thing with thicker and thinner parts, which is like putting a terrible lens in front of your ultrasound. It will cause the ultrasound wave to go in all sorts of directions. So therefore, you can compensate this with this in order to get a focus that you would like. But let's come back towards fabrication or assembly of matter and fluids. And so we're going to try and now use the patterns and manipulate matter to arrange it in space. And so the idea is we start with a microparticle suspension. We will have our hologram together with a sound field generation capability that will then assemble our material into a shape, like the bicycle here. And then what we could do is we can, if we have, for instance, a cross-linking mechanism, or we can fix it in space, then we can fix this whole thing, and then we have made our object. So let's start with not bio, but particles. So here again is an example. This is the phase you need in order to generate this bicycle in solution. The dimension is about something like 4 by 4 centimeters, and the pressure will be from 0 to 100 kilopascal. And this is how you can here now use positive acoustic contrast particles, and in one shot, you can bring them together into the shape of a bicycle, right? So this is one-shot assembly of matter. This is our answer to the DARPA project, right? You don't want to be making cars anymore. Let's make bicycles, and let's do it in one shot. Of course, this is not a bicycle that will ride, but you can take it and manipulate matter. We can also then put some crosslinker and shine ultrasound, and then we can take this sample out of solution, or we can even do it multiple times, and then we can make these letters here by ultrasound assembly. But yeah, letters and bicycles is probably not an industrial need for it, and the potential really lies with biology, right? So could we be taking cells in a suspension, we don't have to push them through a nozzle, and can we arrange them in the shape we like in solution? And here comes the problem, and that is the so-called acoustic contrast. And so if you take a force that is exerted by the ultrasound wave, it will depend on how big your object is, and it will depend on the intensity of your ultrasound, and it depends on this contrast factor. This is the difference of the material property relative to the environment, okay? And so we see here some contrast factors. This is a log-log scale, depending on some parameters like the density and elasticity, which play a role here. Sound speed is important. You see that things like aluminum and silica have 0.6 or 0.5. PMMA, polystyrene is up there, has a positive contrast factor, which means it will, if you have ultrasound intensity, it will be kicked out. It doesn't want to be where there are pressure changes. And then on the opposite end of the spectrum is an air bubble, which has a negative acoustic contrast factor, and it will like to go where the pressure maxima are. And it's huge. It's 5,000. And that's why we use it in the interaction with ultrasound. And the problem that I talk to is cells, because cells sit almost at 0.04. So that means any forces we want to exert are going to be extremely weak. Okay? And why is this? It's understandable because the cell, from a physicist's point of view, is a bag of water in water, right? Very little contrast. So that's the problem. Nevertheless, sometimes it's good to go to the lab and do an experiment, even if you don't think it will work. So here are cells, and they are now in a hydrogel, so they're happy. And also this hydrogel has the ability that it can become harder, i.e. we can fix it either by temperature or with light. So once we've assembled this, we can keep the structure. And the experiment was done by my former postdoc, Fischer, and then we were completely blown away how well this works. These are human colon cancer cells that assemble in solution into a spheroid in an absolutely beautiful way, and it shouldn't happen, right? The physics tells us it makes no sense. And why does this work so well? That really is surprising, and it opened up a lot of possibilities because it seems like we can manipulate cells when in fact we shouldn't be able to exert enough forces. And we can repeat this, and here's the Pac-Man now. Pac-Man cells, right, assembled in a normal Petri dish with cells. These are cells that are being assembled, and here's a line to give you an idea. This is a length scale of 200 microns, so we can make 200 micron lines here at about 1 to 2 megahertz by pushing the cells in position. By the way, we never were able to print a Pac-Man because it turns out that some company owns the copyright of the Pac-Man. And so unfortunately, we were not able to show this in our publication, and my postdoc had to chop this image in half, and so nobody knows what the other half means, but you know now that it's the half of Pac-Man. Moral of the story, always check your copyright before you do your experiments. So We can assemble cells and it works very well. Why does it work very well? And the reason is actually even at low megahertz regions, you are absorbing a little bit of the ultrasound intensity, which means you are causing very slight fluid flows we heard yesterday, beautiful lecture, also streaming effects. So basically you can manipulate the fluid. So what happens here is that in the first instance, the pressure waves, the intensity, will cause fluid flow, which will wash along the cells and bring them towards our weak trapping sites. And once they're near the trapping sites, the acoustic radiation forces can set in and keep them located. And so in this case, the fluid flow is very important and helpful in this whole process in making this work. My title says 3D. I've shown you 2D. So let's try and ask the question, can we define a pressure pattern in 3D? And this is a really complicated problem because it's an inverse problem. I'll explain to you, but this is one problem. And the other problem, almost everything that's interesting has a positive acoustic contrast, like cells even, which means they should get kicked out wherever we have intensity, ultrasound intensity. So how do we solve these two things? Now, we're going to now need multiple hologram fields to overlap them in 3D space. Otherwise, we're not going to be able to realize a complicated object. And even then, it's really difficult to know how to do this. Because imagine you had an object here, and you want to define your object by, let's say, 100 voxels by 100 voxel by 100 voxel. It's a very primitive object now. We'll have 1 million voxels. We will need many, many more to do interesting things. So we have 1 million pressure points in space here, and each one of these will have something like 20,000 points where we can define the phase at each pixel on the hologram. The phase we can do in, let's say, 10 steps. So we have something like 200,000 independent variables, 200,000 independent variables, and 200,000 independent variables that all need to have the right values in order to produce this pressure pattern. So if you iterate this, you very, very quickly arrive at astronomical numbers, and it becomes really tricky to do this by any sequential method. So the first step, how you might get close to this, is let's take a simpler object. Let's take an icosahedron, and let's take the edges, these are the red little spheres here, and let's try and get these assembled. All right? So we have points. And with a point-like object, we can apply a 3D Fourier transform. So we can take this object and we can Fourier transform it, and that will give us a spatial frequency spectrum. And so basically, we can project onto a sphere what we would need to, and once we have this wave defined in the sphere, it will propagate in and give the Fourier transform again, which is going to be this object. But the experimental difficulty is you don't have a spherical sound generation source where you can define wave at any one point. So we need to take apertures, i.e. we make cuts in the sphere that correspond to the size of our transducers. And then we can take 2, 3, 4, whatever, and see what we get. And we get this kind of thing, which will have some interference pattern, but will be an icosahedron. So you can see the same trapping points. So even with 2, you can get this object, but because of throwing away some spatial frequencies, you will, of course, have some reduction in the fidelity of the object. So here's the idea. 2 transducers. And by the way, this is how we solve the positive acoustic contrast problem. These are traveling waves. They want to kick this out at high intensity, but gravity is going to want to pull this down. So the forces in the direction of propagation of the waves are going to counter the gravitational force. Therefore, we can use this method to also manipulate matter with positive acoustic contrast, which is most things. And so here's the experimental setup. So we have 2 transducers, and we have a chamber, and we fill it with particles. And then we want to see that our 3D hologram is going to trap this. And so here is the chamber. We place now everything is in water. This is the hologram surfaces. There's water in here. And now we place the chamber, which is also filled with water, with our particles. And the particles have been shaken, and they can now slowly sediment. And as they're sedimenting through this, they will reach the points where we have defined our acoustic intensity, and in this particular case, in the shape of icosahedron. And you can maybe see that these points are realized here. So we have generated a 3D object via a 3D hologram. We think this is actually the first true 3D hologram, but that's something that we can have a debate about over tea. So here is a point-like object realized with acoustic forces. If you were to switch the transducer off, everything would fall apart. And here is to show that indeed we have, as theory predicts, our trapping sites. This was done with silica gel particles. We have positive acoustic contrast and traveling waves. That's really important here. And to show to you that this is not a fluke, we can take some other platonic surfaces and we can take the edges and we can realize them as well. But how do we go from points to a connected object? That's where we want to be, and how do we fill the volume? And now we need to do this rather complicated computational problem here. We need to slice our object, and we need at each slice have a pressure distribution that corresponds to the object we want to generate. And for this, we are using our Angulum spectra operator. This is basically a propagation operator that takes into account that the acoustic waves have a k vector, and they also have a direction. And we project our sound field pattern then in each plane. Then we need to do this for the other transducer and possibly for a third one. And then we are combining all of these. And now our Angulum spectra operator is in 3D. It's basically a sum, superposition. And then we need to set in a minimization procedure, because now we have a projected field pattern in here that will be our object, our Pac-Man in 3D, if you like. And then we want to, for getting there, we need to optimize it. Therefore, we need to compare it to our target field, and we need to minimize some loss function. And typically, it's sufficient to take a square error type loss function. And then we need to go back and change the phases in our starting position, and then we start iterating, right? Which updating at each point. And the nice thing is we can benefit from the machine learning community, because they have very fast implemented gradient methods in their optimization, which are in things like TensorFlow. And so we are using their approaches. We're not learning this, but we're using their very fast algorithms that are very good in taking derivatives. I don't know if you know this, but with these kinds of algorithms, you can differentiate any function, complex functions, all of this kind of thing very easily, and that's kind of implemented in there. And for the experts here, we are working in the complex plane, so we need to also work with a different kind of algebra. But this is a detail. We are basically taking a gradient descent method with a squared error function, and then we're iterating this. And we, on a small cluster or workstation, in several hours, we can compute simple objects, and this was implemented by Heiner Kremer. GPU acceleration, 3D angular spectrum methods, and the details don't matter too much. And so here's our target now. We want to make a helix. A helix is 3D. We have 3 holograms. They are projecting their pressure fields into space, and this is what we should be getting. And this is what we have in terms of intensity. We can calculate, it came up yesterday, the Gorkov potential. It's a chance of basically figuring out what the forces are likely to be on the object. And this is our hydrophone measurement of the experimentally realized field. So we can really do this in 3D. And here is the experiment, some silica gel particles. And in a normal epi, this is in solution, nothing special, no special container. And you can see this is rotated from different directions, so you believe that this is really 3D. So this is a 3D assembly in space using pressure fields that are shaped. And it doesn't matter whether you take a plastic container or whatever, because the ultrasound will just go straight through. So it's very, very versatile for that method. And here are some more shapes with hydrogel beads and even cells, which we can then take in a gel and assemble it in one shot. And how far can you go? This is Armadillo from the Stanford 3D image scanning repository. And we can fill the volume, but then we wouldn't see anything. So we've made points on the surface. We've oxalated it. And this is what slightly higher frequency at about 3.5 megahertz with 3 transducers we should be able to realize. So this is what you should be able to do with very modest ultrasound frequencies. So you should be able to define arbitrary objects. And of course, the name of the game is to assemble things like multicellular objects, which is another talk, if you like. So time is ticking, and I want to now switch and extend this and have a way of doing something by basically initiating things in space with our 3D pressure pattern. And for this, we're going to do acoustic paint. So you can manipulate objects like bubbles, right? This is what people do, even in a medical context. You can use it for contrast, and you can also burst these bubbles and thereby open the blood-brain barrier, etc., etc. But these microbubbles that are used are rather limited in what you can do other than bursting them, because these little bubbles are just bubbles, therefore they're not very stable. They need high intensity in ultrasound to burst, and if you want to bring something, you can only bring whatever you can load onto the surface of a bubble, which is very limited. And so I'll tell you about some really different ultrasound contrast agent, which is, normally we have gas surrounded by a thin layer in fluid, and I'm going to tell you about the anti-bubble. And the anti-bubble is a very peculiar object. It's a liquid drop surrounded by gas in fluid, okay? And the nice thing is we have the gas, so we have a strong interaction with ultrasound, and we have a core that is loaded with a liquid, so it's like an encapsulated particle, if you like, where we can do a lot of things. How do we do this? It's an emulsion-based method using Pickering emulsions to stabilize these, and this is where we are collaborating with Albert Pottinger. So this is basically an oil and water emulsion with fumed silica, which is going to be our stabilizing surfactant or nanoparticle, and you basically apply this twice to vigorously stir this, then you get droplets, and then you again vigorously stir. So then in the first instance you got water surrounded by silica particles as a stabilizer in oil. Then you do this whole thing again, and you get this in water, and then you get this oil droplet surrounded by water, again stabilized by silica particles. So that's how we can encapsulate something in the center and have it stable in a fluid. Now there's no air here yet, and this is where the real magic happens. You can freeze dry this whole thing, you can release the oil, and then you can put it back in water. You can put it in your shelf for months. You put it back in water, and it will actually bring the water inside in the core here. So this way, and this is absolutely stable. It's really amazing. And this thing here is now our anti-bubble. It will have a water-filled or liquid-filled, it could be your whatever, your drug, your biological agents, surrounded by a fumed silica cell with air, so we can couple very nicely to ultrasound, and everything is stabilized, and this can be put in water. So here are some pictures, tunable from easily between 10 microns and hundreds of microns. And the nice thing is, and this is very, very different to standard ultrasound, by changing the shell composition, you can change whether this pops at very low intensity in a gradual way, or it doesn't pop, and at some point it will start releasing its material. And the intensities are order of magnitude lower than what you need in normal ultrasound. So it's much, much more general. So this is, we think, is really exciting. Here is a super high frequency, fast recording of one anti-bubble in video to show you what the release mechanism is. So this is filled with dye. And unlike a conventional microbubble, you don't just explode it. What happens is that there is some interesting nonlinear deformations on the surface, and you start to shed your shell, and as you shed your cell, you see it becomes lighter here, and you are releasing your material. And this shedding process, you can basically tune. So therefore now, you have the ability to bring more material and release it at lower intensity. And so here is now how we're going to do acoustic paint. So we want to have, wherever we have intensity, like in the form of the psi, we want to have something happen, i.e. release of color, right? And we're going to load antibubbles with dye, and because the dye is very highly concentrated, it's quenched, we don't see it fluorescing, but when this ultrasound is projected and we are at the right threshold intensity, we can release the inner parts of the antibubble, and thereby we can paint where we are. So we have an instant recording of our pressure distribution. So here is what we calculate. This is what we measure. So it's always a very nice comparison between the experiment and the theory. So this is the actually measured pressure with a hydrophone. And then we can take a container, for instance, we make a gel, we load it with antibubbles, they are dispersed, we don't see them, apart from a few speckles. And then we can tune our intensity to be at the right threshold, and we can use a hologram that projects this psi, and then with one pop, we can release our cargo payload in a particular shape. So this gives us the opportunity to use the hologram And we are working hard on making projectors for brain and for releasing them in particular locations, opening the blood-brain barrier and having these interesting carriers to bring things, in addition to assembling matter in 3D space. So I would have had one more topic, but I think my time's run up. It's 35 minutes, so I will probably stop here, right? So that we are more on time. And I was going to talk to you about how to do this dynamically, but I'll switch through this very quickly. And I will end in here, thanking my co-workers and my funding. And I, of course, thank you for your attention. Thank you.
Thanks for a very fascinating talk, Professor Fischer. Almost seemed like magic. Now the floor is open for questions.
Thank you, sir, for the questions. So I have a set of questions that firstly I would like to ask that what are these hologram plates made of? Like what is the material of the hologram plates?
Fully commercial, printable plastic. In our case, it's called VeroClear.
Okay.
Nothing special. You just need to know the sound speed and then you're in business, and you need to make sure it's fairly homogeneous. If you have a printer that is not so homogeneous, then you have scattering in this material that you want to avoid.
The next question is that, so when we are projecting the ultrasound from the hologram plane into the fluid volume, so the fluid is contained in some kind of a chamber. So while the sound is passing through the walls of the material of this chamber, do you expect there to be some kind of a phase change or a change in the direction of the wave vector as it passes through it?
No. So, I mean, you choose your materials that there is good matching between the fluid and the material, so you have very little reflection. And typically, we also line the container with absorbing material so we don't get multiple reflections. But this is not really a problem. Yeah, I mean, it does change its phase, but if the material is a plane parallel structure, it doesn't matter, right? Everything has the same phase value, so it will not deviate the beam.
Okay. And along the same lines, so does the size of this cavity play a role here? Like
it's not a cavity, right? Not a cavity. It's a traveling wave. No, it depends on your wallet. If you can buy a cheap transducer, you get one that has 2 centimeters diameter. If you spend, I don't know, more like several $100, then you can get also large transducers. And that is the only limiting factor.
Thank you very much for a beautiful talk. I love these 3D holograms that you're doing. So you showed us the assembly of cells, and it was partly supported by the acoustic streaming to bring them into the trapping position. Now, when you do this in 3D, you don't have a surface boundary to help you with the streaming. So can you sort of help me how that comes?
From the transducer, right? No, in this, okay. Yes, but in the 3D, the material is brought because it's everywhere or it's falling down under gravity. So it's passing through the trapping sites, right? So then we have material delivery, and we are actually also getting rid of things that we don't trap, which is usually a problem in these cell culture things because the bottom of your Petri dish is full with stuff you don't want. In our case, it just goes right through.
Okay, so gravity is helping you.
Gravity is helping us, and it's also needed because otherwise in a traveling wave you couldn't keep a positive acoustic contrast material there.
But you could then basically apply another field that help you to drive.
You can also do that, yes.
Yeah.
That would be another way, and you could even induce a little bit of a flow, and sometimes we're doing this in order to clear the volume.
Okay, thank you very much.
So a very interesting talk. How are the shapes of the holograms decided for a particular geometry?
So you start with your target image, right? So you have something in mind that you would like. I don't know, you want Mona Lisa in 3D. And then you need to define some intensity like you would in a color image. And the intensity corresponds to an amplitude. And then you need to go through this iterative process, right? So then you need to say, okay, I want an amplitude here. What phase do I need there? And this is done by the 2 boundary conditions. One boundary condition is in your volume, the amplitude, because you know you want to have intensity or no intensity or a little bit or more. And on the other boundary condition, is that the amplitude comes in and it's all the way the same because it comes from a transducer which has everywhere the same amplitude. And so
it requires several iterations to get it right.
Multiple iterations.
Oh, okay.
In a 2D thing, we need about 10 to 20 iterations, and you really it's very easy. It's fast. Any computer, any laptop can do this in minutes. In 3D, if your object becomes complicated, then this will require much more computation power. And it's really the memory, because you have to keep track of these millions and tens of millions of positions and locations and variables. And for this, we need really memories in the range of 100 gigabytes or more for an object that's sort of on the range of something like 2-3 centimeters by 2-3 centimeters, that kind of thing, yeah.
Okay, thank you.
You saw two different types of bubbles. One is like the normal bubbles and the other one is anti-bubbles. So how are these bubbles stabilized? Like, first of all, for the ultrasound agents, are those microns in size or those are sub-microns in size?
No, submicron usually doesn't couple very well. I mean, it does, but even in medical ultrasound, these tend to be in the range of several microns, 5 to 10 microns. And you also have to, of course, keep in mind, if you make this thing smaller, you can also deliver less. The normal bubble is either stabilized by a surfactant or a lipid. Lipid is generally what is used most often, and sometimes people use polymers, but people are not liking polymers very much anymore in biology, so very much lipids, and that's why it's unstable. And in the anti-bubble, it's basically the fumed silica, which are the nanoparticles, which is stabilizing the interface. That's the trick. That is the stabilizer that comes from this Pickering emulsion. Right? And this is very, very stable. It even survives freeze drying, and in solution, we can have this for weeks. If you use microbubbles in ultrasound, it will be injected, it will be circulating for 10 minutes, 20 minutes, and then game's over.
3 holograms, how did you define the loss function? Loss with respect to what?
So you have your target, your Mona Lisa, and that means at every point you know what the amplitude should be like, and you know what you're generating. And so if you are close to this, the loss is small. If you're not close to this, the loss is high. Therefore, you need to update the values to get this particular amplitude.
But we don't know the object.
Yeah, you do. I mean, when you go in the lab, you say, today I want, I don't know what, I want a picture of Pac-Man, right? Then you know what your target is, right? Or you want to make an ear, or you want to make an organoid, right? You want to make an organoid, you say it must be 1 centimeter, oh, that's a lot, 5 millimeters long, and it must be this shape, then you know what you want, right? And once you know what you want, you encode this into the holograms.
Okay. So from 1 transducer to 3 transducer, so what is the difference in performance?
I'm sorry,
I mean, you have 1 hologram,
yes.
Then you have 3 holograms.
Yes.
So what is the difference in performance?
The difference is that, of course, you have more variables, so therefore it's a little bit, I don't know, you may remember some undergrad experiment. You can make a light hologram where you encode the information and you can cut it in 2 and it still works. But the amount of information you carry has reduced. So what we do is we have more variables, we have more information, and therefore we can encode a richer wave pattern, which means we get sharper edges, better contrast, and more agreement with the object you want. So the more waves you can overlap, the more likely are you going to hit the perfect realization in 3D. But because we also need to conserve momentum, it makes sense for us to work with 3, because they come from 3 directions, x, y, z, which gives a total vector pointing against gravity, and so this is sort of almost ideal. And then in the experiment, at some point, you don't have any space. So we have generally now worked with 3.
Okay, thank you.
Thank you.
I have a quick question. So apart from the number of transducers, is there any other parameters that you want to adjust if you want to control? Let's say that you talk about the Pac-Man and you want to create the structure out of particles. And one is about the definition of the boundary, and the other thing is how closely you pack these particles. So what are the variables that we have to control the structures?
Yeah, so the variables you have, of course, one is the frequency a little bit, and the higher the frequency, the shorter the wavelength, the better the resolution, right? So at 1 megahertz in water, you are roughly at something like a millimeter and a half. And if you go to 2, 3, 4, 5, you get into the hundreds of microns regime. So one variable is definitely the frequency. The next one is the intensity. As the frequency goes up, you also increase the streaming. So you will have more transportation of fluid. That might be good or bad. And then you have the intensity to increase basically the force on your objects in the trapping site. And that's one big difference to normal bioprinters. Normal bioprinters don't get you the cell density that are biologically relevant, right? They are orders of magnitude off. So basically when people extrude ink, it's a very, very sparse collection of cells. Here we are right at this level. So if you count the cells in a little piece of material, if you push them together acoustically, you are already at the levels that are relevant for biology. And so for this, you need strong forces to keep them in place, but not too much, because you don't want to cause large fluid flows in this volume.
Okay, thank you. Right, if no further question, thank you very much, Professor Fischer, again, for a wonderful talk. And as a token of appreciation, we have...
Thank you very much. Probably the other side. The other way is better. Yeah, all right. Thank you very much. Thank you. Thank you.